Okay, there we are. So welcome everyone. This is Algorithms of Bioinformatics. Today I'm going to show you a little bit what the lecture is like, and we'll do a little deep dive on one specific problem, and then in a way we'll restart next week from the beginning with a bit of more principled background. But I wanted to flip it around a bit so that you have a good glimpse of what the module wants to do. We'll come to that, and the administrative part I'll try to go through relatively quickly. Okay, this was also the room with the HDMI cable where you have to be really, really careful. Also, if I want to show, change of plans. I'll leave this for the sound later. Okay. In the first parts, I'll tell you a little bit about the organization of the module, some fairly straightforward things, but then also about assessments and how we'll conduct the module. So let me just repeat what I said before. The goal for today is to show you what the module will be like, and then we'll restart a little bit in terms of the material next week. But for most people, I think for everyone, this should be an elective module. I think it didn't exist for a while, so nobody can have this as a compulsory module. If you're here, then because you want to. And I want to make sure that you're here because you want the thing that's actually coming and not something else. It's always a bit hard to tell from the few sentences on the module handbook. Okay. And we'll talk a little bit about assessments as well. So hi again. I'm Sebastian Wild, the German version. The module is going to be in English because it's a master module. Of course, you can also answer any questions or ask any questions, answer any exam questions, etc. in German. Our tutor will be Mohammed, who was here. Saw you earlier. I'll do the tutorials. We'll talk about that later, and Mohammed Omar will look at the submissions of your exercise sheets. Most of the, not at all a copy-paste error, you haven't seen this. This is obviously AlcBio. But the link is also on the Elias course that you will have seen. As always, I'm trying to get you connected outside of the lecture as well, outside of the class where we meet anyways. And we'll use this tool CampusWire for that, where you can join. You need this 4-digit PIN to join. Otherwise, the link is just public, but you also have the link and the PIN on Elias, so don't need to remember it now. You've already seen Slido. I'll come back to that. And as always, as mandated by the regulations, the final exam is the final mark. And there's formally at least no other option. To be admitted to the exam, you have to do the exercises, and it's the standard kind of policy. Looking at the size of the group or the size of the cohorts, I'm leaning towards oral exams, but we'll decide that in a week or two. Okay, that was the module in a nutshell. In a bit more detail, some of the bits. Slido, many people may have seen it before. The goal is to keep you awake a little bit more than you would otherwise be. And we'll use it for two separate things. One is, I'll show you an example in a second of asking polls to the audience, so you can tell me a bit where you are. But you can also use it to ask questions during the lecture. You can raise your hand or just shout it out. That's fine as well. If you prefer not to, you can ask anonymously on Slido, or you can just type it on Slido, whether you use your name or not. That's another option. It's a little less disruptive, and I can cover cover the material I wanted to cover and then tend to questions. I'll try to be attentive to the Q&A on the side, but I'm not always good at that. So sometimes if I miss your question and you feel the point has passed where it would have made sense to discuss it, and maybe feel free to point it out. So let's test this briefly. There's the tools that we use in this lecture, the first three, and there's some AI tools that I'm just curious, so I included them. Nothing else really behind the question. The other benefit for me from Slido is I don't have to count heads. I can just count votes, or can let Slido count for me. That's what computer science is all about, letting technology do the job. And because people can join on the live stream, so if you haven't noticed from the infinite text setup at the beginning, if you must, you can watch from home and join on Slido. It's always nicer if they're a larger group here on site, but still I think on balance, the benefits of having a live stream is larger than of not having it and forcing everyone to be sleepy and in the room. So that's an option you have. So 16 might roughly be the number, 17. So people are well aware of Elias. That's good. Everyone has used it. Would you know about the meeting today, the lecture, if you haven't seen it on Elias? I guess you can see it on Marvin. The Elias is not needed. But you probably don't survive long in your studies if you've never touched Elias, so I guess. And everyone has used ChatGPT, but not so much some of the other tools. That's whatever you want to make from this. None of the above. Okay, I was mixed up the two. That would have been surprising. What are the LLM-based chat systems? Do you want to shout out which did I not include? Grok, XAI, Perplexity, yeah. I guess nowadays any kind of web search has some sort of AI built in, but it's a bit limited. Other big ones? DeepSeek. That's right. Yeah, that's right. Never really used the Alibaba ones. Anyways, there's a bunch more, and there's new popping up every day. It's going to be fun to watch this again in a year or so and laugh at, oh, that was the good old days when this was still around. Okay, let's focus. The important thing was that you know Elias. The other thing I should have pointed out is Slido and CampusWire is somewhat well known, I guess, from other modules that I was doing, but also half of you don't know it. And so I'll briefly talk about what these are and what we'll use them for. They're all fairly intuitive. You'll find your way through it. But let me talk briefly about the material in this module before we spend more time on organizational bits. It's a bit of an unusual module, at least for me. It is still an algorithms module by and large, so a theoretical computer science module. But all of the questions that we'll look at have some motivation from computational biology, bioinformatics, biology, depending exactly where you want to make the cuts. So we'll have to dip the little toe into biology, especially some lab techniques and some molecular biology. I called it backdrop, which really just means it's there in the background for the scene, but it will not be the main focus. We won't cover a lot about biology itself, and I'll try to keep the sources that we use somewhat informal and non-scholarly. So people who study biology would probably not be happy with some of the simplifying versions of that. I think for the module here and what we need to know to use the algorithms, it's going to be good enough. Is a fine line between being sloppy and imprecise and being approachable. So I'll have several sources, and I'll rely on you to also point out if you have others. We'll come to that next week in more detail. I want to stress that the main material that we'll do is your standard kind of algorithms theory lecture. So we'll define problems. There's going to be a part before the definition that motivates why do we do this. Then there's usually a disclaimer box saying, oh, and now we simplify from all of these problems that you would have if you solve the actual problem from the data that you get from wet lab experiments. You have to make some simplifications to start. And then in this abstract model, we can decide what algorithms to use. We can prove things. And at the end, we might come back and try to see how big is the gap between what we can achieve and what a biologist would want. And sometimes the gap is not as big as it first seems. And sometimes it is a sizable gap that's left. And some of these things are evolving. It's still a relatively young field, bioinformatics, with some results fairly flexible and young and still moving. And sometimes people come up with better ideas. I hope that's roughly the picture. If you have questions, you're of course just shouting out or put them on Slido. So we'll survey a bit what algorithmic contributions have happened in the past to computational biology, and hopefully equip you with the tools to contribute to those if you so choose. We'll learn just a little bit of biology as much as needed, as is needed, but not much more. Although I'll point you to kind of as a digression, whenever I work on this, there's so many interesting things to learn, because this is about life and us and everything. There's a lot of exciting things that are well worth knowing and learning about, but you won't have those on the exam for this module. So you can dig into that as much as you want. And I think we should use the group to kind of have some fun together in exploring some of these topics. The module will not go much beyond the very basic things we need. Okay. And yeah, another key goal, as I mentioned before, we'll take a step back, make simplifying assumptions, define some nice mathematical problem, and then try to solve it. You should keep in mind where the limitations of these things are. And the goal of the module is also to make you sensitive for these kind of limitations. Preliminary plan is a bit too much, so I'll probably have to kick out one topic. But we'll see. Maybe I can give you a little bit of each topic instead of kicking out one. We'll start today with this little example of one puzzle that might come up in such a setting. And then we'll look at how to find patterns in genomic sequences, which are just long strings there's no commas, there's no points, no spaces, and still they have some meaning. How do you figure out what that meaning is? Then we'll work our way to comparing sequences. What does it mean to be similar in that sense? We'll look at how can you, from these lab techniques that don't give you at all a possibility to read your genome, how can you still compute it? There's a lot of computer science techniques behind this. And then we'll look a bit, take a step back, go back to some basics from computer science that we'll need for the last bits, specifically searching in strings and indexing strings. And we'll apply that to searching in whole genomes. And then there's a few two other topics that are a bit separate from the rest, just to show more of the breadth of bioinformatics. RNA is another type of molecule that serves roles in your cells, and expresses genes, and phylogenetic trees are about the tree of life, how close are different species to each other, and how can you compute those. Okay, that's roughly the plan. As I said, maybe one of these topics will have to go. I'll try not to skip one entirely. Okay, assessments. We'll have a final exam in one form or another. Oral examination, I think, is nicer in master's modules. This is more flexible, but this will mainly depend on the numbers. Most of the material is relevant for the exam. There will be some specifically marked parts that are just for your interest, that go deeper than I would want to ask you about in an exam situation. What is included, though, is everything we do in the tutorials, and I'll come back to that in a bit. For this lecture, I've also made some slides that have a green background. That's whenever we talk about the biology part. And for that, I guess it's only superficial knowledge is needed. You should be able to explain the problems that we worked on. So you should make sense of the narrative, but I won't quiz you on the specific names of some chemical compounds, or I don't know, what does DNA stand for? This is not what I'm interested in the exam. Okay, to be admitted to the exam, as I said, standard rules. We'll have a weekly exercise sheets. Well, the first one will come out today or tomorrow, and then you have a week to work on it. So next Friday is the submission. And then at the moment, we have a slot on Fridays, which will start in 2 weeks, where we meet and discuss the solutions. It's also, again, this lunchtime slot, which I think is not great. If in a week or two, I'll ask around if it's possible to move that to a more convenient slot where all of you have time and we find a room that's large enough, then I would suggest moving it, but you'll hear from me about that. If not, then it stays on Fridays. And so no exercise class tomorrow, nothing to discuss from this week. No exercise class next week because you'll just be working on your first sheet. In the third week of classes, we'll start. Okay? The exercise sheets themselves, they'll pop up on the public module website, the hand-corrected link from the first slide. You'll submit it on Ilias in groups. Try to make it not more than 4 people. I think 3 would be better. If the group size stays like this, I think that should be manageable for the marking. Standard kind of things. I'm not insisting on students presenting solutions in the tutorials, but it's encouraged. I think it's nicer, instead of hearing the same person talk that already does the lecture talking. It would be nice to hear some other voices, and we'll have an eye out for submissions where you've done a good job, and then we would ask you to do it. But if you don't want to, I'm probably not going to force you. Okay. As in master's modules, I tend to see the exercise classes as an offer for you to engage with the material. And the requirement of these 50% of the points is intended not to be a threshold that you cannot pass, but rather an encouragement to spend some time with the module. So we'll mostly mark and give points for sensible attempts at the question, even if you don't fully solve it. Okay, it's different from a written exam where the marking would be stricter. I think that's good enough in this case. It makes our job for marking also a bit more efficient, but it means you're a bit more responsible for your own learning. You can probably make your way through with group submissions and so on without having done much and still get the exam admission. But the exam you'll have to do on your own. So don't cheat yourself, right? Okay. Also some slides about AI stuff. We talked about it earlier. It's fast evolving, and these tools are really cool, but they can't really help you learn. And the more advanced the modules get, the trickier the distinction becomes. I think the tools are also not as good with the advanced exercises yet, at least. Still, basically don't deprive yourself of the opportunity to solve some nice puzzles. If you're sitting in this room and you don't at all enjoy puzzling over some math questions, maybe the module is not the ideal choice, just to be honest. Many of the questions we study and many of the problems in the exercises will have a sort of a puzzle component. And the fun really comes when you enjoy these kind of things. You can also work your way through if you don't enjoy them all that much. That's fine. Try not to destroy the puzzles by using AI on them. Specifically, it's okay if you use it for background research. And for this module in particular, asking it some generic questions. How does this whole protein synthesis in cells work again? I think this is amazing use cases for chat assistants, because you can adaptively quiz about the parts that you're interested in and then stop when you have had enough. It's not okay if you do major parts of the assessments, if you don't acknowledge what you've done. Just don't do it. Be reasonable. Same, of course, applies to copying other people's work. It's not just about plagiarizing AI stuff. Hope that goes without saying at the level you are. You have to work things out yourself, or at the very least, if you copy someone else's work, means you cite it, then you put a proper reference and say what you did. I think I put this on the exercise sheets. If you feel like you're better working in a larger group on some exercise problems, then do so. But every group has to do their own write-up, and you should acknowledge who you've worked with. Just for this kind of, we're moving more towards a scientific mindset. So the culture of proper credit for who achieved some fact is important. Give proper credit and cite people that you worked with. Let's do this quick one to wake you up. I know this kind of gets old. If you've seen it before, then bear with us for 5 minutes. The idea behind this is if you anonymously ask people various character traits or various life situation criteria and properties, and also as one of them whether they self-reported as cheating on some assessment, then you can do a correlation analysis. What kind of property? Maybe if a person, okay, well, what can I pick that's uncontroversial? Hardly anything, I guess. A person that prefers blue pullovers is more likely to cheat. It's just a correlation, so you don't know which causes which, right? But there was one thing in some studies that popped out as strongly correlated with whether people were more likely to cheat. And that's what I'm asking about here. And I want to give it a positive spin, but I'll let you think for a moment. I've hidden the answers so that you're not too influenced by what the others already wrote. I'll show them in a bit. Okay, there's some fun ones. Let me just show them. And there's a bit of an overlap between people in this module and other modules I've done where I used the same question. Yeah, it's never about cheating, right? It's about getting caught. Laziness. They sit on the last row. Okay, so the kind of right answer is in a way among them. Ask ChatGPT. That's an interesting one. Are people that heavily use AI tools more likely to cheat? I don't know. So the study is too old to tell. I think this is an interesting one. I'd be surprised if the correlation was that strong because 100% of people in this room reported using ChatGPT. So it's you can't get much signal. Comment?
I mean, technically, there is already a study that shows that doing a lot with ChatGPT, well, you unlearn to think. So basically, if you encounter hard problems again afterwards, you will at some point have to ask ChatGPT because you forget how to think about the problem yourself.
Okay, the comment was there's a study apparently that shows using ChatGPT heavily makes it harder for you to solve problems afterwards on your own. That's not implausible. I don't know the study, so don't know what they've looked at in detail. That's not unreasonable to expect. I guess it depends a bit on how heavily you use it and so on and so forth. But yeah, so in this case, AI use was not the focus. What they've pointed out was whether or not people procrastinate or self-report to suffer from procrastination was the #1 predictor. So the fact that you spend time on things where you in hindsight think, oh, this was stupid, I would have wanted to do something else, combined usually with the deadline-driven mode of working, that you push things because you procrastinate on doing other stuff that's much more fun, or maybe just, let's not psychoanalyze why people do this. But it leads to then having to do it last minute. And I guess it just means once you become desperate, you start looking at all options, even if you know that they're not good or morally wrong or whatever else. Which also means the best way to get out of this problem is to better manage your time so that you don't fall into the last minute desperation, and ideally don't procrastinate all that much either. It's a bit easier said than done, but there's simple tools to better manage your time. I'm always tracking, for example, what I work on, not so much because I'm interested in what I spend my time on, but when you have to click what you now want to spend your time on, already is a bit of an incentive not to do something different. And there's many small things like this. It's not really part of computer science, but it's part of life and growing up and so on. That's my little kind of nudge. Think about whether you have a system that works for you in this respect. Let me go very briefly over the other tools. CampusWire is essentially a Reddit for lectures. You can ask questions, you can answer other people's questions, you can upvote answers and questions. The main distinction is that you can ask things anonymously, and it's a kind of closed space just for this lecture. Plus, you can write things in Markdown and use LaTeX formulas, which is just a nicer way to do it compared to other forums, whether or not on Ilias or elsewhere. It supports, I would mostly want you to use public questions for anything that other students might be interested in. Use a public question. Only really use the private questions when it's spoiling some assessment or it's really, really something that only concerns you. Most of the things should be a public question on CampusWire. So in a way, when we're here together, use Slido for immediate questions or find me after class. For any time outside of the lecture slots, if you need anything asynchronously, the default will be CampusWire. If you send me an email with a question that I think other people would also have something from the answer, then I will just redirect you to CampusWire anyways. Okay. Otherwise, I think it's relatively straightforward to use. There's also a chat room feature that you can use, but for the lecture, I'm not going to heavily work with it. Let me just reiterate why I think CampusWire is worth the hassle. I know it's another account, it's another thing to check. You can set it up so that you get emails if someone posts a question, so you don't have to monitor it or use their app. I think it's important that everyone gets the same answers if you ask questions. That's what I don't like about office hours. It's fine for some personal things, but then if it's a question about the material of a lecture, I'd rather have everyone see the same answer. And I want to nudge you into teaching others. If you help others understand the material, that's the best way for you yourself to understand it. And by answering other people's questions, that's what you can do. So the only thing that really means is I'll redirect you to CampusWire by default. If you ask a question where I think some other student might be able to answer, I'll not answer right away and give other people a chance. And if they do answer, I'll endorse the answer or augment it if there's some parts missing. So even if it's some other student answering, you'll know whether you can rely on the answer. Any questions on this? There was one question on Slido. Might as well talk about this now. Is the module classified as theoretical or practical? Does it have a formal meaning? I think it's a theory module. I don't know if the module handbook has a specific binary property. The thing is that also the updated module description I couldn't yet find. Even though I wrote one a year ago when we updated the new exam regulations. I think what we have on the website is still the previous one. By all intents and purposes, I would consider it content-wise to be a theoretical module. Everyone knows Ilias, everyone loves Ilias, so I'll restrict usage to the minimum I can, namely everything that has to do with marks or with personal information. If on CampusWire, if you want to sign up with a fake name, that's fine, right? I don't need to be able to identify people. It's nice to know your names a bit, and so you don't have to go anonymous. But if you prefer not to use your real name and uni address, that's all right. Ilias is the official thing. Anything with marks will go there, and the rest is more informal. And whatever I can, I will put on the public website. That's more convenient, in my opinion. Let's talk about this another time in more detail. I want people collectively collect questions for possible exams. What these will look like depends on the exam, whether it's oral or written. And you probably won't start right away anyways. It's just a Google Doc that's linked on the website. And it worked in previous modules quite nicely, at least towards the end of the lecture when the exams loomed a bit closer. I think it's a useful resource to help you prepare. And over the years, we can keep that collection growing. More on that at a later point. The last thing, the physical copies are there in the front row. Feel free to have a look. There's no required reading or so. Everything you need will be covered in the lectures, but sometimes it's good to hear something explained from another perspective. And books are really good if they're good books, and these two are ones I like. We don't cover nearly all of what they do, and there might be some bits that neither of them really cover in that way. There's some other sources that I'll point out. On the module website, you'll find a subpage for each unit with some other links. But these are the main two things that cover most of the material in, as I said, slightly different way. Now, especially the first one here is a very modern one, and it is available as a book, but their main thing is an interactive online version of the text. Now, they're in the US, where it's standard for people to buy textbooks, and a book of this sort would very, very clearly be above $100, and it would be required for everyone to buy it. I'm just saying this to put into context why they charge for access to their website. It's a different system. It's not a very nice one, and I'm not going to encourage you to spend money outside of this. The books should be available via the library. At least the biological sequence analysis we already have, and for the other, I'm trying to get one ordered, where you can also speed this up by asking. Still, the interactive online text has a lot more than what we'll do. In particular, it has lots of programming exercises. If you want to go a good deal more into this and maybe involve yourself in more practical questions of this sort, I'd recommend it. And they shared some discount codes that I'll put on Campus Wire for people to check out. Maybe just give it a try and see if it helps you. It's very, very nicely done and has lots of fun extra side stories and so on, much more than I have time to tell you. It's just one lecture a week. Okay. With that advertisement, that's all I wanted to say for this intro part. It really leaves just 45 or 40 minutes for the little puzzle, but we'll start. If you have questions about the organizational part, we should do that first, though. And then we'll do a 5-minute break and then do some real stuff. Any questions? All good so far? All clear? Okie doke.
Front. Front, left.
It's not very loud, is it? Okay, let's continue. There's a little bit of of a puzzle that I want to show you. I picked out one that doesn't need really much biology background. This really has just the inspiration from biology, and then we take a big step away from it. There's a chapter in the Bioinformatics algorithms book that goes into the same problem, but then tries to solve it from a more practical perspective, and we'll stop short of that. And given the time, I'm not even sure we'll get to the full theory solution, but we'll see. Still, I think it's a nice example because it's a good puzzle, even if you don't buy into the biology motivation. And it's a good illustration of the kind of things you'll see more of in this module. This first unit is really just an exemplary example, a tiny puzzle almost taken out of context for showing you what the module will be like. And we'll take a step back next week and do some of the background in a bit slower pace. But for today, I want to go straight into this. The puzzle we'll look at is essentially sequencing proteins, where here this means reading what the protein is made of. And I'll tell you a little bit of what proteins are to start with. And then we'll look at the abstract problem that's behind this. That's the turnpike problem. Okay, so what is protein sequencing? Well, for that, we should know what proteins are, I guess. Proteins is a whole class of biological molecules. And in a way, they're among the most interesting ones that we have. You may know protein as a nutrient, so meat is protein, and many other things we eat consist of protein, and our muscles are made of protein, and much of our cells are made of protein. But they can actually do a lot more. First of all, what they are long chains of little beads, and the beads are amino acids. There's more chemistry behind how the chain is formed, but you could really think of it as kind of a chain of pearls, which are connected with a very strong piece of string. So they're really hard to break in that chain connection. They're very stable. But also these little circles that are drawn here, they're not all the same. These amino acids come in many different colors or flavors if you want. At least 20 are the standard kind of variations. And they have different properties. For example, some of them are electrically charged or magnetic, and so they attract each other, which means in water, as these mostly exist in our body, they fold into three-dimensional lumps or knots or shapes. And here you see this is the red and blue part. These are two different proteins, but they together form this almost knotted structure, and that's hemoglobin, which somehow manages to capture oxygen. I don't know if the green thinks that's not oxygen, it's way too big. Okay, don't ask me about the chemical details of how hemoglobin does this treat of capturing oxygen and then moving it via your blood cells, red blood cells, through your body and then releasing it at the right time. I'm a bit out of my depth here as well. The point is that proteins are essentially a linear shape, and if you know these, the colors of these beads, if you know the sequence of amino acids, then many of the other functions that the protein has can be worked out from there. Or at least you can try to make these proteins synthetically if you know the sequence of amino acids. The structure is usually, the function is determined by this three-dimensional structure, which is yet another question how to get there. There's a few things listed of what proteins do in our bodies. It's a wide range. Our hair are proteins, so they can be fairly stiff and really stable structures, but they're also working as enzymes. The chemicals that break down food, even in your saliva, and there's hemoglobin, as I mentioned. Some hormones like insulin are proteins. The sequencing of insulin was one of the Nobel Prizes won for proteins. One of the few people that were twice awarded the Nobel Prize. And the first he got, I think this was, okay, I'm not going to say wrong things. There was a Nobel Prize for sequencing the amino acid sequence of insulin with obvious applications for all the diabetes patients. So proteins do a lot of things, and that means in bioinformatics, there's many things we want to know about them. We want to know the amino acid sequence. There's a long puzzle that's kind of solved of how to get from the sequence to the three-dimensional structure, where AlphaFold seems to work well enough that people are using this to at least guess what the structure will be and what the function will be. They interact, as you can see here, so there's lots to study about them. Too much. For us, we'll just focus on this problem of determining these amino acids in the sequence. To make this a bit more precise, there's also these unpronounceable names. These are the standard 20 amino acids that you usually find. They have a name, they have a short code. For us, they're just part of an alphabet. So a protein, its sequence is a string of these letters. So every letter is one amino acid. And because there's too many, usually you need a slightly longer name than a single letter in our alphabet. And they have different chemical properties. There's the sum formula of what they consist of. But of course, the structure is also important. And they have different molecular masses that we'll come back to soon. Okay, so much for proteins. The key question is, for this unit, if you're given such a molecule, you know somehow it exists in some cell, how do you get at the sequence of amino acids? And now let's completely forget the problem of how you isolate this specific protein that you want to look at out of this messy environment with all the million other substances. Suppose you already have a way of isolating that. How would you get to the sequence? The, in practice, most successful option is actually a completely crazy indirect one, which comes from the fact that most proteins are made by your cells according to a blueprint which is written in your DNA. And so if you read the DNA, it's actually easier to work out, simulating that process, what the protein will look like, than actually measuring the protein itself. It sounds nuts to me. And the reason this works is just that our technology for doing the latter is so much better. Inherently, it seems much more complicated. But there's some proteins where this also doesn't work. If you learn anything about biology, then it is that every rule is broken in some example. For whatever weird thing you can come up with, you'll find some example of some weird creature that evolved to do something different. And for proteins, that also exists. There are some that are not made from genes. And maybe at least for these, or also because you want to do it more directly. There's also lab techniques for sequencing proteins if you just have the final product. And a very versatile one is mass spectrometry, where essentially you take the molecule, you break it into lots of pieces, and you try to measure how much they weigh, the different pieces. Let me try to play a little bit of this video. Now you're not going to hear much from it. That's the problem, because I couldn't get the sound really to work. Isotopes. And it's a bit not too bad, but okay. So you can watch the full version at some later point. So that's what these things look like. We don't need the history for this one. But let's look at the explanation of what this machine does, because I think it's useful to have a little bit of at least a sketched idea of what happens to appreciate what we need to do afterwards.
Move electrons through the sample. So there's a little cathode ray tube. It produces all these electrons. What it's going to do is it's going to pull electrons away from the sample. And then as it does that, it's going to create a number of positive ions. And so we're going to ionize that sample inside here. Remember, it's still the sample, it's just ionized. It's lost its electrons. And so now we move to the mass analyzer. That's really only going to have two parts in it. It's going to have an electrical field. You can see that's negative because we want to...
So already before that step, what he's showing down here, it's a little cut off. Let me move it up a bit. Well, what you see down there in this box is already your protein, many copies of it, essentially broken into pieces in all possible ways. You have so many copies and you break it vigorously into many pieces that it essentially breaks at every possible position. That's going to be at least our abstraction. And then the chemical parts that are left over, they'll have protons in the nuclei of the atoms and they'll have electrons. And you try to, don't ask me how to do that in detail, you somehow get the electrons away with an electric field. So what's left are positively charged particles because they lost the electrons. And so that you use in what comes now.
Move the ions into the mass analyzer. And then we're going to have a magnet. And what that magnet is going to do is it's going to bend the path of the ions. And so as we're bending the path of the ions, it's just like driving around a corner. If you're really heavy, it's hard for you to make a corner, let's say if you're in a big semi-truck. But if you're in a little motorcycle, it's easier to make it. And so this is where we're going to figure out the difference between the mass of those ions. And then finally, we have a detector. That detector is going to be made up of two things. We're going to have an electron multiplier, which is essentially a plate. As an electron hits it, it spawns more electrons, which hit the next plate, which spawns more electrons. And so we can really have a small amount of ions or anything hitting that plate, and we're going to get a signal. Now that signal has to be amplified, but eventually we can send that into a computer and we can look at the spectrum coming from those different masses. And so the first thing you have to do is you have to calibrate the machine. What does that mean? You're going to start sending ions through. Okay, so that ion didn't hit the detector. Why is that? It's because the magnet is turned up too high. And so we're going to have to lower the strength of that electromagnet. We run another ion. Okay, now the magnet's not quite strong enough. And so now we run another ion, another ion, another ion. Okay, so we've calibrated it, and so it seems like it's working well. Which of these would be heavier? Well, the ones that are heavier are going to be the ones that can't quite make the corner, and so they're going to end up out here, and then the lighter ions are going to end up right here. Now let's actually get to some sampling. And so this is what it's going to look like when we create a spectrum. We're going to have the different weights across the bottom, and then we're going to have the intensity. And so wherever the intensity is high, we're going to have peaks. That means we have a lot of ions that are with that specific atomic weight. And let's try chlorine. So we're going to put chlorine through here. Chlorine really only has two stable isotopes. It's going to have chlorine 35 and 37.
So he's doing a much simpler example. It's a single element, not a whole protein. But it turns out even a single element is not just a single variety. They come in different isotopes. And so if you send this through, there'll be different peaks for the different amount of neutrons in the core. Really, there's different weights. Maybe that's enough of that. So you get this even for very simple chemical compounds. One thing to keep in mind is what you measure here is not actually the atomic weight of the particles. It's a ratio of charge and weight. That's one problem, because you can have ions that lose several electrons, then they have a larger charge. And so they're more influenced by the magnet. That's one of the things we're going to ignore entirely. And amazingly, you can do the same process with much larger molecules. You can still reliably break them into many, many pieces, and you'll get these huge number of peaks for all possible little parts in that molecule. That's basically what the lab technique can do for you. Take many copies, break them in all possible ways, and measure how much of these different parts you've got. Okay. So much for that. We'll abstract from that a bit, but even at this point, it might sound totally crazy to reconstruct this linear sequence of the molecule from just these peaks of weights. And the way to rescue us here is that you will get many peaks also for segments of the whole protein that are still intact. So you get substrings, and you get the sum of the weights in that substring as another peak. And I have to somehow work out from the collection of all peaks what the sequence was. Let's take a step out of the lab, and in this section do the mathematical formulation of the problem, where we can reason about what exactly that is. Now, here's another, this is just a fun one. Do people still know this guy? The problem's called the turnpike problem. And I wanted to at least show where the name comes from. Do you know what turnpike is? If you've carefully seen it here, it was already in the right. I think it comes another time. So I'll go back briefly. Sorry, it's too late. Wow, okay. Amazing series, fun to watch. Sopranos, about a mafia gangster having regrets, and there was the New Jersey Turnpike. Okay, I'm not gonna show it again. It's essentially a toll road. Yeah. It's a toll road. It's the American name for that. A turnpike is really the gate that people in medieval times had, where maybe someone would charge for letting you on their road that they maintained. And for some reason, it still to this day is used for ordinary highways. Whatever. The problem comes from this name for the roads, because what we'll think about is a toll road. You can have many exits. It's a linear arrangement. It's a one-way highway. It's a little stupid. You can enter the highway at any of the exits. That sounds wrong. Any of the, well, they're still called exits, right? You can get in at any point, and you can get out at any exit. And essentially what you pay is proportional to the distance you traveled. Now, suppose someone just tells you they've driven a lot, and they just give you their credit card statement with all the charges from the toll road. Can you work out what distance was there between all the exits and in what arrangement they are on the map? That's why it's called the turnpike problem. Let me properly explain what the problem is. Compared to the mass spectrometry we've seen, we simplify a few things. First of all, we'll assume all the molecular weights are just integers. And we'll ignore this nasty bit that there's these isotopes where for some things you have slightly different weights, even though they're the same atom or the same element. We assume every breakpoint that you could possibly have. Remember, your protein was this long chain of beads. This long chain of beads, and you imagine you have copies broken in every possible way. I wanted to just draw this picture from before again, but maybe it's good enough you can imagine. So there's one copy where you broke it between those two, and for any other possibility, you get a peak. And we also assume if there's several ways to break the molecule into different pieces so that the sum of their molecular weights are the same, then we know this. well, you'll see this in the spectrometry spectrum by having a larger peak. But we assume that we can exactly read off how many copies will have that peak. And we'll also assume that nobody spat into our sample and contaminated the whole thing. With that, we can assume our input is a sequence of numbers. That's the beads from here. Each has a different amino acid, so each has a different weight. Okay. That's our input. That's what we, in the end, we want to find this. But for the moment, that's what's given. That's the sequence of numbers. The first thing we'll do is we'll do prefix sums. So we take the sum of the weights from the beginning up to a certain breakpoint. That's just for notation. So S for sums, P for protein or positions. Or I guess let's stick with protein, but maybe points. I don't know, it's the distances. And then the key definition is the difference multiset. So that's all the numbers we can get from going from the ith to the jth position in our sequence. And all combinations, but it's sub-words. It's not arbitrary collections of beads. It's what we could get by breaking the molecule in various places. Okay? And it's a multiset because it could be that some of these numbers are the same. So a multiset essentially says you can have several copies of the same number. And we know exactly how many of each. Because we do that, the number of these entries here is fixed. For every pair of position I and j, I get one entry. I'll have some examples for you in a sec. Let's do the examples first. So here's, that's the beads have weight 3, 5, and 1, and 2. Now you can break this up in arbitrary ways, right? So you could have just the 3, or the 3 and the 5, or 1 and 2, and all these possible options. Formally, what we defined was the prefix sums, and then take differences, but you could really just work out. We have the 1 from the one molecule that's just bead with weight 1. Then we have a 2 because there's a bead of weight 2, and there's no other way to get a sum of 2. But we have 2 copies of 3 because there's a bead of weight 3, but also the substring of 1 and 2 sums to 3. Okay? And so this is, if you do this for every possible I and j, they go from 0 up to n, where here n is the length here. N would be 4 in this example. And then you sum up exclusive. So, okay, from I to j minus 1, pk. That's the sums that you get. And then 11 is the sum of all 4. Okay. Here's another example, a simpler one. If all the weights are the same, they're all 1s, then you just get, from all possible combinations, you get 5 times the 1 from picking each of the 5 individual elements. Then you have 1, 2, 3, 4 options of getting length 2 by every pair. And you have a few 3s and 2 4s and a single 5 for the sum of all of them. Okay? Question?
Why do we don't have 2 times the number of weeks in the first example? Is the order of the...
The only subwords. Yeah, that's important. That's a good point. So there's a 6 because we have 5, 1. But you cannot take arbitrary subsets. Let's maybe, for this small example, write down all possibilities. I think it doesn't take long. So we have the 3, the 5, the 1, the 2. Then I have 3 plus 5. Nobody can read this. I have 5 plus 1. I have 1 plus 2. Then I have the 3 plus 5 plus 1. 5 plus 1 plus 2. Oh God. And the sum of all. And that's all. Okay? That's why this is, if this were not the case, there would be no way to get back to the sequence of numbers. You need to somehow have some information about the order. But all we've got is these weird partial sums. And then the turnpike problem is the inverse problem. We're given these multi-sets of differences, and we're supposed to come up with a set so that if you build this difference multi-set, you come out with the input. And so here's an example where this doesn't work. It's not always possible. First of all, the difference multi-set has to have a sensible number of entries, otherwise you're immediately out of luck. But here 3 is fine. You get a size 3 difference multi-set if you have 2 elements. So just 2 beats. Then we get the differences, take the first beat, take the second beat, and take their sum. And if the difference multi-set is supposed to be 3 times the same thing, that's not going to work. Right? Then whatever you put, A and B could be the 2 numbers, and it'll never work out to that distance subset. So it can't always work. There's a question on this. Are we missing subsets of size 1? So sequences of length 1? That's a nitpick of my definition. They're in there. Let me go. Maybe it wasn't the smartest way to formalize it. It's nice and quick to write. So here we take the difference of 2 prefix sums. And I'm not allowing I and j to be the same. That would give me a difference of 0. But I'm allowing j to be I plus 1. And so when j is I plus 1, I get essentially one of the pi's. You could also have written immediately just some of these, but then you would somehow need a notation for summing up a substring. I found this smart, but maybe it's more confusing. All it says is what we've done in this example. You have the 4 letters. Now you can take the length 1 substrings, the length 2 substrings, the length 3 substrings, and so on, and always sum up their weights. Okay, the room fell silent. That's the right moment to have you think about a bit. This is the first non-trivial size, right? It's 6 numbers. So n will have to be, I'm looking for 3 beats. Then I can make 6 combinations, 3 length 1 substrings, 2 length 2 substrings, 1 length 3 substring, and that's it. What weights would give me this difference multiset? We're heading for a cliffhanger lecture. I'm gonna postpone the. Yep. So you have the multiset here. So that's that one. Yeah, I think few people started figuring it out, and then one submission somehow got split into 3 answers. I don't know if you can fix it. because the example is so small, you can essentially solve this one by trial and error almost. We will have to have the length 1 substrings somewhere in there. And there's no way, okay, it's integers to make things even easier, but there has to be some weight 1 beat in the chain. Otherwise, we don't get this 1. And we have a second 1, so there's going to be 2 beats of weight 1. All right, so there's a 1 and another 1. That's part of the ingredients. Now, this 2 here might be the sum of those 2, so maybe they're adjacent. But we have another 2. Probably that 2 is another beat. So a guess would be that the 2 are adjacent, and then the 2 is the last one. And then you can work out there's going to be a 3 for these 2 and a 4 for the whole thing, and that actually matches. That wasn't too bad, maybe. Various people found that in the small case here, you can do it by trial and error. But it's kind of hard, right? Yeah. That's true. That's true. Yeah, the whole problem is oblivious to reversing the word. So the best we can possibly hope for is identifying the sequence or its reverse. We cannot distinguish those two. Yep, that's a very good point. Let's spend a few minutes on how we could systematically solve this. And maybe you already get the feeling that this is not going to be an efficient algorithm. The name already suggests this. And to not kill the exercise, let me explain the intuition maybe in more detail next time, and first show the algorithm. The key idea is as follows. You know that in the difference multiset, you're going to have the contribution for the entire string, summing everything up. And because the numbers are all positive, that's got to be the maximum. So this 18 here will be the sum of all the weights. Can't be in any other way, and it also has to be a unique maximum. Otherwise, we can immediately say it's not possible. So we can always say there's a starting point at 0, and there's an end point at the maximum in our difference multiset. So far, so good. So the 18 we've already dealt with. That was easy enough. Now how to continue. And the key idea to have is that from the remaining difference, there's again going to be some maximal value. And that's going to have to be the next longest substring that we sum up. Or apart from this 18, which is the sum of all the beats, the next maximum must come from one that has 1 less beats. We don't exactly know which and how it looks like. That's all complicated. But there must be some other distance between 2 points of length 17. Yep.
Maybe I got the definition wrong, but the leftmost number must certainly be one of the beats. So basically we can subtract that from the rightmost, and we have at least one of another beat that will certainly be sum of multiple beats. I mean, of course, this number can be there twice, but one of those will be a sum.
The tricky bit about your suggestion is I don't know exactly which of these numbers are single beats. But there's one where I do know, namely the smallest. The one has to, is that what you meant? Okay. Okay, the leftmost number. Yeah.
Must be 1 beat.
So these two together are kind of the next step we can make. The 1 is a single beat, 17 is all the rest. So there's only really one option, but But how to place them? The beat 1 could be at any position. So the beat 1 corresponds to some distance here where the difference between the endpoints is of length 1. And where exactly that belongs on this line is a bit unclear. The 17 helps us in that there's some point between a start point and an end point that has to have length 17. And that's the longest. So the 17 can either start here and go all the way to here, or it can be the other way around. But it cannot be anywhere in the middle. Otherwise, you'd get an even longer one. So there's just two options that we have to check. And then the algorithm says, well, I don't know which one. Let's try both and see if we fail or not. And you can work this through on this example where it does get to a solution. But let me show the code first, because that's what the exercises pick up on. This is exactly that idea formulated in pseudocode. There's nothing else going on, but I want to make sure that the code is clear. So it's an algorithm that gets the difference multiset as an input. It starts by picking out the largest entry there. That's the total distance. And then it starts guessing a position. This should be rather called S, I think. It plays the role of these cumulative sums. So the collection of ticks, locations where I have made a position, and these are essentially the cumulative sums because I start at 0. So the naming is a bit unfortunate. We know initially we start at 0, we end at this total distance, but we know nothing else. And then we have a recursive function that in each case, maybe we're already done, we've covered all the distances, then we're happy. Otherwise, we find the next largest that we haven't covered. So we take the total difference multiset and we subtract everything, all the distances that we get implied by the current points that we've already chosen. And among those remaining ones, we pick the largest. And by the same argument, it has to be either flush left or flush right. And so you try these two options. And flush left means you add this largest as another point. That corresponds to picking this one. Then we had a tick here at position 17. And so we add this D to our point set. But we don't add it right yet because we don't know whether it works. What we do is we check if it already gives a contradiction. Namely, if it implies the current point set implies a distance multiset that's not contained in what we have, then we're doomed and we cannot continue. Otherwise, we have to keep trying. And then we do a recursive call with this slightly larger point set. We might still at the end come back. Yeah, there's a check missing. If it's not this no difference multiset. You only return if it succeeds. If it fails, then you keep trying the other option. And that looks symmetrically. You take the distance between the largest and this D and try again if that worked. And if neither of the two options work, then it cannot work. Yeah.
Why don't we know that the first two parts of the set are definitely, because if there's only one thing, it can't be the sum of each other, and the second thing can't be the sum of the first thing.
I guess that's right. It's just not clear immediately what to make of that second piece of knowledge. So you're right. What we do implicitly uses that the smallest number has to be the weight of some bead. And it's also correct that the second smallest weight also has to be the weight of some bead. But we don't know what to make of that. All we can use is placing beads somewhere. And the second smallest I guess could be almost anywhere. I'm not sure if there's an easy argument to improve upon this. But keep thinking. Yeah.
You know, next to each other, it's like the first some of each other.
Do you know where to place it? Maybe we can discuss offline.
Not definitely.
Okay, good to see that people are hooked by the puzzle. This algorithm, the last thing to say, we discussed the correctness on the example. The bad thing about the algorithm is that it's an exponential time in the worst case, because you try two things, and what you want, what I would like you to puzzle out in the tutorials is that you can indeed make this go really bad. It tries things for a long time and still fails. And the overall problem, this turnpike problem, is unclear whether it's NP hard or not. It has all the flavor, all the feeling, the smell of a hard problem, but we don't have a reduction, so we don't know. Okay, with that, let me leave it. There's a little but coming, but that has to wait for next week. See you next week. No lecture, no exercises tomorrow. Keep an eye out for the tutorial sheets. I already told you essentially what you'll do. I'll patch the code to not return immediately here. And then we see each other. Make sure the register and campus wire. There's nothing else to do on Elias for now. And I'll send some information about this online book round.